class antibody, we get a lot of dying cells, and you can see the ones that are green and red are dying myeloma cells. When we treat with another inhibitor of a downstream pathway, we get some green and red. But when we combine these, we get many more, 40%. We can increase the efficacy of these treatments. So, is it murder? Murder is not effective. We need to induce the cells to commit suicide. Another thing that Dr. McHale was emphasizing was the effect of the microenvironment. So it takes a village. One of the most important things that we've learned over the past several years is that the tumors don't grow in isolation. They don't stand on their own very well. And they need the stromal cell, the stromal cells that are represented here in gray, and the myeloma tumor cells. There's a give and take, a very clear interaction between the myeloma tumor cells, which are secreting growth factors that are activating the stromal cells, which then secrete growth factors that support the survival of the myeloma tumor cells. Excuse me. And so it takes a village. These cells don't exist in isolation. The tumors don't grow in isolation. Not only do they need their friends, they need their environment. So the underlying matrix that supports these tumor cells impart important signals. And you've heard NF-kappa B several times, and we found that not only do mutations within the tumor cells activate this NF-kappa B pathway, but signals that are derived from the extracellular matrix also activate this survival pathway. So location, location, location. And finally, as it's been emphasized, myeloma is a multifactorial disease, and one of the best ways that we're going to treat this disease, we're going to find therapies, is through combination. And so this is just an example of a study that we published last year in which we're measuring DNA damage in this assay. And so if we use Velcade only, we get a little bit of damage, which will induce cell death. If we use this topo2 inhibitor, we get a lot of damage. But if we can use Velcade to increase the target of the thonophyte, we can get a dramatic increase in cell death. And this should be very specific for the myeloma cells and not overly toxic because of the specificity of Velcade for multiple myeloma cells. And this is all in bench-based lab work, but I believe there is a clinical trial that's based loosely on this principle in which Velcade is combined with a liposomal doxorubicin. So where do we go from here? We have to continue identifying the new targets, which John's working very hard to do. We have to invent new therapeutic agents, which means that we have to talk to synthetic chemists, and we have to collaborate with people that make things like this. We have to devise rational combinations, and we have to develop new models. And that's been one of the huge challenges in multiple myeloma research from the bench perspective, is that we really don't have good mouse models. Mice don't get multiple myeloma. So I just, a little bit of nerd humor, when the big mouse is telling the baby mouse, stay away from scientists because they cause cancer. And so that's exactly what we'd like to do. And I'm going to show you a little bit of unpublished work of a model that we've been developing at the Arizona Cancer Center in Tucson, that we can test therapeutic agents in mouse models of myeloma. Now, as you know, myeloma is a disease of the immune system, and as you've heard today, it relies heavily on the microenvironment. And so we can't really use many of the standard models that are used for solid tumors. We can inject the cells into the flank and measure the growth, and it may mean something, but it may not, because that is not the environment that myeloma lives in. So we started with a multiple myeloma cell line, it was derived from a patient probably 40 years ago, and we carry it in culture in the lab, in flasks, and as long as we give it all the nutrients, it will continue to grow and divide. And we took the gene from the firefly called luciferase, and this is what makes fireflies light up. And we put this gene into the multiple myeloma cells, and then we injected those into a mouse. And this is just the initial experiment, just to see if we could detect the light. And what you need to take home from this picture is these were injected subcutaneously in the left flank of these mice. And then the mice are put in an imaging system which collects the light that's emitted by the firefly luciferase that is inside the myeloma tumor cells. And this is essentially a heat map, and so the blue is cool, and as you get towards the center, it gets hotter, if you will, or brighter. And so you can quantitate the amount of light coming from this tumor that's implanted in the mouse and measure the tumor growth and test drug activity, because if I give these mice Velcade, it goes away. I'm sad to say that's not what happened here. These guys just didn't grow tumors. So as you can see, it's not entirely reliable as far as tumor growth. And that's probably because the flank doesn't really provide the right environment. Well, so this was just a test to see if we could get the cells to grow and if we could detect the light, and it worked. So we moved on to the next step. What we needed was a model that we could get the myeloma cells to grow in the bone marrow. But how are we going to get them there? And how are we going to know that they're there? How are we going to monitor the tumor cell growth once they're in the bone marrow? Because as any of you that have undergone a bone marrow biopsy or aspirate knows, it's not a pleasant experience, and